Hi guys, we will be going through the mass transport question pack. So let's get started. So we've got here question number 1, and in here you were asked to work out the percentage error. The equation was given, so uncertainty in given measurements divided by mean multiplied by 100. However, extra follow-up information it says that uncertainty in measurement is half of the range. So to work out the percentage error, we first of all needed to work out the range. So the numbers that we have taken for this range would be the 9.3 over here, take away 7. And it says half of the range, so we're dividing that by 2. We're getting 1.15 divided then by the mean. And if we work out the mean, it's 8.2. Multiply that by 100 according to the equation, and we're getting our final answer of 14.02%. Finally, the next question was asking to use the understanding of the mass flow hypothesis to explain how the pressure is generated inside the foam. So remember, foam transports sucrose, and to transport sucrose, obviously, we do it through the process of translocation, and anything to do with the pressure, it's using the fact of the water entering from the xylem. So here I've drawn out a little diagram to remind you that sucrose is actively transported into the foam by companion cells that now will lower the water potential in the foam. Hence, the water is going to move by osmosis from the xylem when there was obviously higher water potential. Okay, this is where our answer is coming from. So sucrose is actively transported into the foam, lowering this water potential, and obviously the water moves from the xylem into the foam by osmosis. Another question here requires you to look at the differences here in the pressure and the changes in the water flow in the xylem. So if we look here quickly at the relationship and at some patterns, hopefully we all can notice that at around timing here between 8 and 10, so over there and over there here, we can see that the pressure in the foam has decreased. However, quite opposite, the water movement in the xylem has increased. Opposite pattern we can notice between around 5-6 o'clock. So here we see the increase in the pressure. However, we can see that the water movement, the rate of the water movement has decreased. And as the question we were asked to describe this relationship, so describe only say what we can see. So this is what we've just seen there. So the foam pressure will fall as the rate of the water movement in the xylem increases. So, so we've discussed those patterns here with the arrows. Okay. Another part of this question refers to the foam pressure being reduced during the hottest part of the day. What we need to remember, now obviously with the hottest part of the day, there is the highest light intensity. So if you look to at my diagram to the left here, the light intensity causes transpiration, so stomata opens. So that increases the water loss through the transpiration, and of course the water is lost through the stomata which open. What does that cause then? That causes the intake of the water through the roots, and water coming in increases the pressure, which we call the tension. So higher tension we've got here as our second marking point, or the water is lost through the stomata. Any of those will be fine, and that causes less water movement from xylem to foam, because obviously we need to deliver the water back to the parts that were lost so the photosynthesis can continue. Question number 2 then. it's mainly based on the practical aspects. So the students has investigated here now the water movement in the celery stalk in grams per hour and per group of xylem vessels. So what I've done here, I have kind of put the units that the students will be working at. So grams, obviously referring to the mass, per hour, referring to the time, and per number of the vessels. So that should help us out with answering the questions. Another important bit of this setup here, we've got the layer of the oil. So remember, here in terms of the layer of the oil, that will be waterproof. So that's important to kind of consider within the past paper questions. And let's have a look here at the questions. So the students measure the time taken of the water movement, and they're asking for other measurements that he should take it. So as we discussed at the beginning, obviously we've got grams. Grams refers to the mass. So the same like you did with the required practical 3 with osmosis, if we want to see the change in mass, we need to record our raw data of initial and final masses. So raw data, remember, it's a data just recorded without any calculations. If you were then to work out the percentage change of that data, then we will call it a process data because we've done some calculations with it. And coming back to our second marking point here, remember number of the xylem vessels, that's another measurement that should be taken to work out the rate accurately. The reason of the adding the layer of the oil, we say it's waterproof, so it will prevent the evaporation, so less transpiration, or transpiration will only take place from that celery layers over there. Question C then, it's another part of the investigation here where the students had cut equal lengths of stalk from each plant, and we've got the whole setup here described of how it has to be done. Intervals, that's something good that the students actually follow here as well. And in figure 2 we've got the shown slice of the leaf stalk with colored water inside groups of xylem vessels. So quite frankly, like the water is lost obviously from the leaves, the water also will be lost here from that piece of celery. What does that mean then? That could help us with the explanation why color water has moved up the stalk. Obviously, because the water has evaporated, as we say, from the stalk. So now, if we're looking at the difference here in the water potential gradient, so water potential gradient, we've got lower water potential created, so the osmosis of water will be taking place. So the water will need to be taken back, because by losing the water, we've lowered the water potential here in the stalk. With the water surrounding the stalk, which has higher water potential, the water can move into the stalk by osmosis. Okay, so this is the here the pattern. Obviously, if the water comes in, it comes together as a column because the water molecules cohere, so they stick together by hydrogen bonds, which is our third marking point. Okay. Question D refers to the health and safety working skills which you have gained with the required practical 5. So how do you cut safely? So obviously we need to cut away from our bodies and make sure that we cut against the hard surfaces. Another, we've got the set of the results produced in the table. So we've got the distance measured in the millimeters. And the students have here given us some data, and we needed to decide which of the measurements would be good to carry out using the information given in the table. So whether it's a mean value, which obviously works out the average, median, which is a middle number when we put them in the order, or the mode, the most common number. So if we're looking here, the best calculation that we could do, because we've got outlined number, will be the median. So if we organize those numbers in the correct order, so from 30 to 80, the middle number that we would achieve here, it would be in the middle of 40 and 42, which gives us 41. Question 3, it's nothing else than the model answer, asking you to describe the cohesion tension of the water molecules in the xylem. So remember, we'll quickly here put our poster one more time. So that's our plant. So due to high light intensity, the water is lost from the leaf because of the transpiration. Transpiration now will lower the water potential in the mesophyll of the leaves, and that will allow the water to be taken in by the roots from the soil. So remember, in the soil we've got higher water potential, in the roots lower water potential, so the water will enter by osmosis. Water will be pulled up as the column to replace the water that has been taken, obviously has been lost through the stomatas from the leaves, and it's that water column, it's possible because the water molecules cohere, so they stick together by the hydrogen bonds and adhere to the to walls of the xylem, so they can be pulled up together as the continuous column. So that's a cohesion tension, which explains the movement of water in the xylem. Question 4 here, we were looking at the very difficult experiment, so we needed to investigate the effect of the substance X on the growth of the new roots. Remember, if you ask for the effect, it could have increase, decrease, or no effect at all. So we need to establish what are we talking about. Another very important here information, it's about the ringing experiment. So if you remember through your notes from the reading material, here with the ringing experiments, if you cut off the part of the phloem, so imagine this is the section that you've cut over there, and the phloem has been cut off, the sugars will be accumulated above the cut. A little bit information about the agar in this experiment. So if the students have used the agar plate and put it on the top here, as you can see, of the stem, that means that diffusion from that agar block could be taking place, so it could act as a source. What I've done later on with those setups here, I've color-coded them to show you what are the differences, and I'll suggest to do it because it's shaded where you can see what's different in there. So for instance, D could act as our control because there is no substance X that promotes the growth and the stem is intact. E has a substance, the same conditions remains as in D. F shows you the aspect of the ringing experiment, so the phloem has been removed from there. And in G, we've got the effects of the lower temperature. Obviously, if the phloem has been removed in F, remember, if there was effect of the ringing experiment, we would see here like a bulk, so there should be more sugars accumulated on the top above the cut. If the temperature has dropped to 4 degrees, we know now that there is obviously less kinetic energy. The processes such as respiration can be affected, so no ATP will be formed, and so on. So, and here we've got the mean number of the roots that they have achieved as the effect of those 4 setups. So if you're looking at the treatment D, E, F, and G, E seems to have the most of the effect, the most increase in the number of roots, comparing with D, F and G, it's all almost double. Well, what I'm normally comparing this to, it's D, because we did say D acts as our control. So if I want to see the effect, obviously I would compare many to the control, and that seems to double. So what you needed to do, you needed to explain why the measurements obtained from this control is used by scientists. So obviously what I did a second ago, we've used it to compare effect of other treatments. Okay, and obviously it shows you the measures the effect of the substance X. So whether X does have any effect or not. Question B, we needed to use the diagram and the table to see what we can conclude. And we were looking at those values a second ago. So we've seen that D shows that substance X, it's not required because D doesn't have the substance X and still has the growth. So the number of the roots that we've got in D, remember, it was 5 over there. So still some growth in there. Also substance X, as we can see, moves through the plant because when the parts here has been cut off, okay, so when we're looking here, for instance in the F, this form comparing with the this substance comparing with the setup in D hasn't achieved the same amount of the roots, so there's some missing part here, so it definitely has to be moving throughout the plant. So that's what we will obviously conclude from here. It has to move from the form, as you can see here in the accept mark. And finally in E, we can see that substance X has caused the growth because it has doubled the number of the roots which we've observed, so it's gone up to 11. Question C then, the most difficult probably question in the setup we needed to evaluate. In evaluate questions, we're normally getting 4 marks, so you can see here the full marks, it's 4 for this question, and you always need to put 2 for and 2 against, so you need to be balanced. You wouldn't get marks if you just went for one of the aspects. It has to be a spread here. So what we've got to support this conclusion, as we've said, the form is involved. We explained this with the previous kind of question. We said that if the temperature has decreased to 4 degrees, that will now affect the active transport because there will be no or little ATP being formed. And remember, ATP is formed in the respiration. So if temperature drops below the optimum for the enzymes, that reaction will be affected. We have discussed already with the question that the AGA blocked allows the diffusion of the substances, so acts as the source, and roots are our sink. So those were the supporting arguments, and you've got them in your booklets as well. And let's have a look for the against. So for the against, in the setup F, as we've drawn out before, so I'll remind you here, we would expect over there to see this bulk. Okay, so not meant to be here if it was a ringing experiment. However, that's not visible, so not bulk above ringing in F. So also there was no information about the osmosis and xylem. So not role of osmosis or hydrostatic pressure. Also, what we could say, if the substance X was moving downwards towards the roots, and that meant to promote the growth of the roots, that could happen obviously due to gravity down. Roots still grow without the form, as we can see in the practical D. In D, there was still 5 of the roots. No leaves has been here shown because obviously this agar was acting as the source, and in normal practical normal setup you will have leaves acting as the source. However, we did have roots, which were our sink. So that's the most probably challenging part of this question. Evaluate A of 3. Remember, yes and no aspects are needed. Question 5 then. In question 5, we were looking at the effect of the heat treatment. So quite similar as we have before. So in heat treatment here, we are using our carbon, and they told you that the carbon is radioactively labeled. What does that mean? That means that we can detect it, we can see it, if let's say we use the X-ray film. So in figure 1, we've got the treatments. We've got treatment A and we've got treatment B. So heat treated and in A and not heat treated in B. So if we first of all look at the outcomes of the experiment, and every time if you've got standard deviation, you need to refer to it. And in this example, we do have standard deviation. If we look at those values, standard deviations overlap. So there is no significant difference. However, what's important to mention here? In the heat-treated plants, as we can see, or maybe not see, apart from this point over there, we cannot see any evidences, any movement of this carbon. So we need to be thinking what could happen here. Well, if carbon, obviously, it's incorporated into organic substances such as glucose, sucrose. Sucrose is the one that's transported around the plant in the living cells. So phloem is obviously made of living cells, and heat will damage them. So again, like in previous question, there is no mitochondria, so no ATP. And obviously, transport of the sugars, of the sucrose, does happen by active transport. And if that has been damaged, we would not expect to see much of the transport. And we needed to explain the results. So here we've got a couple of the explanations. So let's start with the ones that we've mentioned. So the reasons why we might not see much in here is because the carbon dioxide, not carbon dioxide, but the carbon, sorry, has been converted into other substances or sugars. Other ways that we've got here, what we've mentioned, is the fact that the movement in the phloem requires living cells, requires ATP, and heat treatment would damage those cells. There was another marking point also here that says the mass flow or translocation, if you like, in the phloem through the plants only taking place where the plants were untreated, so in picture B. So that's something that you could get the mark for as well. However, that was worth only 2 marks. Question B, then we needed to explain how the results in the table support the conclusion. So we did say that obviously phloem is a living cell, so made of living cells, so it would be damaged by the heat. However, xylem is dead, so it doesn't really affect the fact that the xylem has been treated with the heat. So does the table show us the standard deviations overlap here, so there is not significant difference. And the reason behind that is obviously because water is still being transported in the xylem. Xylem is dead, so it's not affected by the heat treatment. We've got figure 2. And let's have a look here at the figure 2. What does it show us before we look at the conclusion? So remember to look properly at the axis. So we've got the ratio on the y-axis. We've got the old and young leaves on the x-axis and the key. Many students are missing on the key, so don't be one of them. Look at the key. And let's have a quick look here. So what I've highlighted with the green over there, it's the effect. So the heat treatment on the old leaves, and the young leaves were represented as the white bar. And what we can see clearly, it has more effect in young because there is a massive decrease. However, untreated then has less effect. As we can see here, the bars don't differ as such. Hence, our conclusion. So, let's have a look then what we've got as a proposed conclusion. So heat treatment has a greater effect on young leaves than old. Well, we've seen that here clearly with the table, so we can tick that. We are happy. So heat treatment damages the phloem. And if we look at that, obviously, it is here the case because that's how the substances would be transported. Phloem is a living made of living cells, so that's the same scenario as a second ago. The ions has moved up the leaf, and if they were moving up the leaf, they were moving in all the plants by the xylem. Okay, so that explains the movement here. There are also other marks that you could be getting. However, there were 4 only, and those were the most friendly marks. What else we can see from the data? That there is a higher ratio of ions in untreated old leaves than young. So that would be the friendly marking point. But also a very common marking point is referring to the statistical test. So if there was no statistical test, so no standard deviation, we don't know whether there was a significant difference or not. And that's always worth a mark on the exam. Okay, so worth remembering that marking point number 9. Question 6, it's straightforward question. Ask you how the xylem tissue is adapted to its function. So you should have used your class notes to answer that question. So moving on to question B. Question B here which shows you the rate of flow in the xylem against different times of the day on the x-axis. So at the time of 2 o'clock, we can see that the rate of flow in the xylem seems to be the highest. Reasons behind that, of course, it's the time when there is the highest light intensity. So if that's the case, the process of transpiration is going to be really high. So that's then our answer. This increase is explained. Remember, explain, we need to say why using our scientific knowledge. So we've got increase in the light intensity, which increases transpiration. So water will be lost because it gains more kinetic energy, as the stomata will also open more often. Remember, stomata opens to get more carbon dioxide for the photosynthesis, but as a negative side of this opening, it allows the water molecule to escape by transpiration. And final question here asking you about the diameter of the tree trunk. So remember, in the hottest day, there is a lot of tension, a lot of pressure, and that it's caused because of the adhesion. So the water molecules attract themselves to the walls of the xylem, and that tension, that pressure allows the water to be pulled up as the column. Okay. Question 7 then. Question 7 requires a little bit of understanding here of what the graph shows. So again, we've got our x-axis and y-axis, and we've got the tension in the xylem. We've mentioned about tension a couple of times. So tension is the pressure. The same like with the water potential. If the water potential becomes more negative, it's something that we need to consider. The same here. So the greater the tension means more negative. And how then that graph works? So we can clearly see here then when the tension increases, becomes greater, means more negative, it's because of the light intensity being higher. And if we look at this, okay, That's where the tension becomes greater, means more negative. The light intensity is also the highest here, so 250, and that obviously later on it will level out. What happens then when we've got high light intensity? It means that the stomata obviously will open, which we discussed that a second ago, and increase the rate of transpiration. Coming back to the aspect of cohesion tension, so remember the water will be pulled up the xylem as a column, and we can refer here to the fact that water molecules cohere together, so stick together, so can be pulled up as the column down a water potential gradient. So that was question A. In question B, what's also needs to be kept, so is it important to keep the humidity constant? Well, yes it is, because if you think about it, the water potential inside and outside the leaves here then would be affected if there was a difference here. So in that case, if we keep the humidity constant, the water potential gradient would not be created. Hence, there will be no transpiration that will prevent the water loss. Question 8 here, again about transpiration, so the water loss, and the student in this scenario has observed the weight of the leaves. So any loss of weight means that the water will be lost. And in one of the setups, in setup B, the student has used the grease on the upper surfaces. So if we put the oil, we did a question with the oil before layer, so that means that will prevent the transpiration being waterproof. So that's the setup they've did, and the next figure shows you the water loss. Again, read through the axis. So water loss as the mass loss has been recorded in time, and it seems like set A, so the one without the grease, has more water loss. However, set B has the grease applied, and we can see there is less water loss recorded. So what environment conditions we should keep constant? So orders that affect the transpiration, so like light intensity, humidity, air movement, and the temperature. So you've got all this recorded in your notes booklet in the table. Student measure the water loss in milligrams. What's the advantage of that? Well, typically what you will measure will be in grams. Milligrams gives more accurate, more precise results here. So remember, think what's the difference between milligrams and the grams? Obviously, large difference so improves the accuracy, gives you precision, and decreases the chances of the error. Explain the change in the maths. So remember, explain, say why using your knowledge. So in A, there was no layer provided here. So there was a decrease in the water loss according to the graph due to transpiration, because obviously stomata has opened and there was a water potential gradient created. Hence the water has been lost. However, after this diffusion gradient has become constant here, the stomata will then close. Question D, asking you about the setup B. Remember, in setup B we had grease. So if you've got the grease layer, there will be no transpiration. Stomata on the upper surface will be covered by that, hence it will remain close. And that is obviously preventing the transpiration one more time. Question 9 refers to the potometer. And with the potometer, remember, it's the practical that allows you to look at the water movement. Not fully explains or evaluates the losses of water through transpiration, because there are other processes taking place in the plants that are using or producing water. However, we can still use it to work out the movement of water, and we will do it in the centimeters cubed. So remember, We get that from the capillary tube following the movement of the air bubble per centimeter square. So this is the surface of the leaf, and per minute, so per time. What's important with this setup, remember we've got the tap, we need to make sure that it's closed properly. We've got the reservoir here, and it's a very important job of that because you can add water and we can always push the air bubble back to the beginning, so back to the red spot if we want to restart our practical. So here a couple of the questions on the setup. So we did say, how do we return the air bubble? Obviously we could open the tap, and that air bubble, let's say if we moved here, it will be pushed back to its original position over there. So other steps the students should follow to make sure that the practical has been carried out properly. So what we've said, seal joints to make sure that our tap is closed, cut shoots on the water, cut shoots at the slant, dry off leaves, so the same like you did with the potatoes in the required practical 3. Make sure that any apparatus is inserted on the water. Make sure that you record where the air bubble is at the beginning of the practical, and ensure that no air bubbles, other air bubbles are present, and obviously shut the tap. And as I've said at the beginning, the potometer doesn't really measure the uptake of the transpiration as such, because the plants are using water to support, using water for photosynthesis, but produce in respiration. And also what could go wrong with the apparatus, it could leak. And the students has recorded the results here in the table, and we need to look at the relationship, so the pattern. So if we look here at the pattern, as the number of the leaves increases, the mean movement of the water decreases. So that's the pattern. We can see that, but that's description. We needed to explain why. So why is that the case? Because having more leaves will lower the surface area, hence less transpiration will be taking place, and as the effect, obviously, we've got less cohesion, and that is explaining here the lower movement of the water, so lower uptake of the water. Question 10, our final question in here, coming back to the movement of the sugars, movement of the sucrose in the foam. So just to remind you, how does that work? So obviously, in the source, sucrose has been here produced. Well, the glucose has been produced in the photosynthesis, converted into sucrose, which will be now actively transported from the source using companion cells into the phloem. So once we in the phloem, the green one represents here the sucrose. Sucrose now lowers the water potential in the phloem, so we get water from the xylem as it has higher water potential, and the water moves into the phloem by osmosis. Any uptake of the water creates the tension, means increases the hydrostatic pressure, causes the mass flow, so it pushes the sucrose molecules to the sink, to the roots. So, and that obviously then removes the sucrose actively from the phloem, lowering the water potential, so the water then can go back to the xylem. So the main sugar, main carbohydrate that will be transported, as we've said, is sucrose. Here, what causes this hydrostatic pressure? Well, we've said higher concentration of the sucrose coming into the phloem lowers the water potential, so the water now enters by osmosis from the xylem due to difference in the water potential gradient. So it goes down a water potential gradient from the higher in the xylem to lower in the phloem. And we've got the radioactively labeled carbon dioxide here. and it has been incorporated into the organic substances produced obviously during the photosynthesis in the leaves in the source. So as we know, obviously the leaves are our sources, and from the leaves we will transport the sucrose to the sink. The sink doesn't have to be necessarily our roots. It could be young fruits, for instance, the growing parts of the plants. So here we needed to say yes or no to random questions, and we needed to back ourselves up with some evidences. So remember, it's radioactively labeled carbon dioxide, so we can detect it and we can see it. So where can we see it? We can see it in the young fruits, in the tips of the roots. Tips of the roots is very important to mention because this is where the growth is taking place. So you should remember this from the required practical 2. So I put the RP2 as the reminder about using the terminology of the tips. Translocations taking place, yes, it is, because as you can see here, the sugars has moved up and down. If it was the cohesion tension, we will be only expecting the movement up. Translocations taking place in the xylem rather than phloem. Well, we cannot conclude that from the diagram because we cannot see the tissues that were used, and the only carbohydrates were transported. Well, again, we cannot agree with this because carbon can be incorporated into different molecules such as amino acids, glycerol, fatty acids, and any other carbohydrates. Right, guys, so that's the end of our question pack. Please make sure that you've got it now completed with all the labels and all the answers. I will be checking that in the class. Thank you.